very kind introduction. I would like to also thank Mariam and Emily for getting me here. It's been so far one of the most really rewarding experiences in lectureships, particularly the time I spent with the students today and the time I look forward to interacting with many of the scientists here. Of course, I'm deeply moved to be giving the lecture in memory of Professor Obaid Siddiqui, although I am saddened that he was not here for me to meet him personally. I've learned quite a bit about him through readings and from hearing from Mariam and others, and he's truly a role model and inspiration to all of us. And I hope this lecture will do some justice to his curiosity and sort of passion to always explore and discover and inspire. What I'd like to do today is share with you the story that got me to move from the clinic to the lab. And hopefully through that story, you will see how important basic research is for us to understand human disorders. And also, of course, how important is catching on medical clues to pursue research. I think the two really interact. So as you've heard, my story started in a clinic at Texas Children's Hospital when I was a first-year intern in neurology, when I encountered a patient who was supposedly a healthy young girl till she was about 2.5 years old, 2 years old, when suddenly she fell silent and lost her social interaction. Prior to this, she was able to say a few words, to sing along in a nursery song, and always to greet her dad when he came from work. But all of that stopped, and instead, as you'll see in this picture, she lost the ability to use her hands. She started wringing her hands. You will see as she attempts to pick an object, she really can't do that. And when she walks, her gait is not quite normal, has a little bit of balance problem, and she developed spasticity and total loss of communication. She also had some breathing abnormalities. I was quite struck by seeing her and looking at all her clinical features and learning of a paper published by Ben Hagberg. It was easy then to match her symptoms to the symptoms of the patients reported in this paper. Ben Hagberg is a Swedish pediatric neurologist, and he recognized the constellation of symptoms in a group of girls along with his colleagues. And he called the syndrome progressive syndrome of autism because they lose social and communication skills, dementia because they normally start with normal developmental milestone and then they regress, ataxia for loss of balance, and loss of purpose handful use. And as he went around Europe describing these patients, somebody pointed out to him that an Austrian pediatrician, Andreas Rett, has reported in this, but in a German language article in the 60s. So in honor of Rett, they called the syndrome Rett syndrome. And our patient had all the features. Now, sometimes in medicine and in science, serendipity probably is a little bit more important than any particular discovery, and that's what happened in my case. A week after I saw Ashley, I saw another patient who also had symptoms of Rett syndrome. And that patient had the typical hand-wringing movements, which is something I had never really encountered in many of the neurological cases I've seen. And she had the autistic features, rocking, loss of communication, and balance problem, and so on and so forth. So recognizing that this must be Rett syndrome and recognizing that an entity that hasn't yet really been seen or reported on in the United States, here I am, in 1 week I see 2 patients, I was immediately inspired to look for more. And I worked with the clinic volunteers and we went through records and quickly found additional 7 cases. So within 1 week we had several cases of Rett syndrome, and I reported in that and immediately became sort of the expert on Rett syndrome, and many patients came and saw us, and I was really intrigued by all of them. The syndrome affects every aspect of the brain, every function, and the best way for me to describe it is think of any psychiatric or neurological syndrome you've ever encountered in anyone, put them all in one patient, and that's what happens to these girls. Every part of the brain is affected. Now, all of these were girls which led me to think this must be a genetic disorder. I could not really imagine any reason why a disorder will only show itself in a female and never in a male, except perhaps if it's genetic. And that's when I decided maybe I should find the cause of Rett syndrome, and maybe if I get to learn how to do molecular biology and molecular genetics, I could find the gene and maybe study it in animal models and eventually figure it out. And this is today what I'd like to tell you about, the journey we took and where we are today. And of course, our ultimate hope is to bring successful therapies to these patients. There was a little bit of a challenge, though, to this, and I'd like to share that with you just to show you how far we've come in science. The challenge is that by now I have hundreds of girls with Rett syndrome, but each of them is, as you see in this family, one girl in a family. So there's really no evidence of inheritance. It looks like it's a sporadic occurrence. And back in the 80s, it was extremely hard to clone the gene for a sporadic disorder because you have no way of mapping it on the chromosome or finding where it is and getting closer to it. So although we saw our first patient in 1983, it did indeed take 16 years to find the gene because of that difficulty, and I had to work on other things in the meantime to keep my career going, and that was an important step. But throughout those years, we encountered very rare families, and here are two examples where you would see that these two affected girls shared a common mother. They had different fathers. The same is true for these two girls. And the fact these two girls shared a common parent suggested to us that this really may be genetic. This gave me more affirmation that it's maybe genetic, and maybe helped us narrow it down from the whole genome, all 23 chromosomes, to perhaps one chromosome, the X chromosome, which would be helpful as you are trying to search for a needle in a haystack. And the reason I picked the X chromosome, and that was the suspicion at the time that this, if it is genetic, it is indeed on the X chromosome, is because the mothers who might have passed these mutations are healthy. They appear normal. And that could only happen if a gene is on the X chromosome for the following reason. You look at the putative carrier mother, you'll see that she has some blue cells and pink cells. In each female, we have two X chromosomes, a red one and a blue one in this cartoon. And as you know, males have a single X chromosome. And as you can imagine, nature will not have it that females have twice of something, that males have only once of one thing, right? We have to be equal. So nature's way of making all of our genes equal on the X chromosome is that in every cell, only one of the X's will be active and expressed. So typically, in any female in this room, a healthy female, you will find that half of the cells will express the gene from the father and half from the mother. So this way you'll have just one complement in gene. And our hypothesis was the reason this mother is healthy, perhaps if the mutation in the gene that causes Rett syndrome is one of the X's, sometimes only the cells with the healthy X will survive and make it, and this woman would be lucky to have most of her cells with the healthy X, and she will not have any symptoms. But if she passes the mutation to her daughters, and her daughters have the typical X inactivation that you'll see in any typical female, which is 50-50, then half of their cells will have the mutation and will manifest. And indeed, we tested for that, and we found this is the case. And that was our first clue how to get about this gene. Then we got these sisters, and we asked, if they got the mutation from their mother, then the mutation has to be in a region that's shared between them. And we've done mapping studies to do that. And it took a while, but we narrowed it down to about 30 million base pairs, which can contain tens to hundreds of genes. And we started marching gene by gene, sequencing them one by one until we find the correct one. And as you've heard, it's a gene that encodes for a protein called methylcytosine binding protein 2. So one point I'd like to make, and this is particularly important for the students, is it took 16 years to find the Rett syndrome genes. And today, if I was to see my first patient with Rett syndrome, and I would like to pursue finding the gene, I will be able to find it in 2 months. Because you don't have to have any more families to map. You can just take a few affected individuals, sequence their genome, analyze the data, and the mutation will immediately become apparent. So this is really transforming medical genetics and transforming our approach to understanding human disorders, where we can identify the causes of so many disorders today in the hope of better understanding. Now, let me tell you what we've learned since the discovery of the gene. First, let me tell you a little bit about the protein. The protein was identified 10 years before by Adrian Bird as a protein that binds a particular form of modified base pair in the DNA called cytosine when it has a methyl group. And the discovery at the time was that this protein interacts with other proteins to silence gene expression, to keep genes at lower levels in our cells. So once we discovered it as the gene causing Rett syndrome, the next thing was for us to understand, does it cause anything else? What kind of clinical syndromes are associated with this disorder? And what is the in vivo function of this protein? And how does it contribute to the disease? How is it that you take that protein from 50% of the cells and now you have all the neurological symptoms I mentioned to you? The first thing I'd like to review with you is when we went back to the clinic after the gene discovery, what we learned. We learned that mutations in this gene can cause the typical Rett syndrome, as you've heard, but it also can cause atypical Rett syndrome, which means milder forms where you only see some features of the disorder. We also learned that in some females, it will only cause mild cognitive deficits, or might cause just autism, or might cause anxiety and obsessive compulsive disorder. So quite a variety in partial symptoms, which was quite interesting. And I'm going to show you a video now of a patient who also had a mutation in this gene. And you'll see she's quite different. She had a much, much milder course. She presented with autism. Here's one who had a lot more spasticity and tremulousness. And here's one who had a lot of movement abnormalities in addition to all the other features of Rett syndrome. So what causes this variability? Sometimes the mutation type can cause that. If the mutation is milder, partially affecting the protein, or more severe. But if you recall, I told you this is on the X chromosome, and I told you there is X inactivation, and that in every female, half of her cells will express one X, and the other half will express the other X. It turns out for Rett syndrome, if you have 50% of the cells with the mutation, you're going to have full-blown Rett syndrome. But if you have just a few cells expressing the mutation, and the rest of the cells expressing the normal X, now you can get these partial clinical phenotypes. And this is important because we now have many patients as such. The other thing we learned since the discovery of the gene is that males actually can be affected, but they have much more severe clinical picture. Because the male has a single X chromosome, every cell in his brain and every cell in his body will carry the mutation. So if the male have a mutation that totally inactivate the protein, that will cause a neonatal disorder, very severe brain malfunction, and the male will die within a year of life. However, if the male has a milder mutation, a mutation that barely affects the protein, just we call these sometimes missense, sometimes at an amino acid, that a very subtle change, then that can give symptoms such as schizophrenia, juvenile onset schizophrenia, bipolar disorder, and autism. So it goes to show the broad spectrum of clinical problems we can see with mutations in this gene. So having the gene is really helpful because now you can try to create an animal model, and the animal model will allow us to see how the disease develops, what can we learn from it, how can we explain the symptoms. So we use mice, and as you know, mice share a lot with human behavior and genome similarity and so on. So when we made a mouse model where we put one of the human mutations in the mouse, we got a mouse that appeared normal for a period, just like the human patients, and then it had learning and memory deficits, hypoactivity, spasticity, balance problems, and so on and so forth, pretty much all the features of Rett syndrome. When you study the mouse in males, they will also have premature deaths, just like the human males. I should mention females with Rett syndrome typically can live a normal lifespan because this is not a degenerative disorder. However, they have increased risk of sudden death, and that's still being studied, the cause of that. The most startling thing was the typical hand wringing we see in the human patients. We were able to see also in the mice. It was reproduced in the mice, as you'll see in this video. This is a healthy mouse, and you can see it keeps its paws apart. And then you're going to see the rat mouse doing the constant hand or paw, in this case, wringing activity. That's really reminiscent of the hand wringing activity. It's a very typical feature of this disorder. So now we know Rett is caused by losing the function of this gene, and we know that in the mouse, if you take it away, you reproduce the same symptoms as the human. The first question we wanted to ask, what happens if we put the gene back? Because that's a sort of step one towards testing the idea of gene therapy. Maybe we can replace the gene, and maybe that would be helpful. So we decided first to test that idea genetically by just adding a copy of the human gene in the mouse with the goal of eventually breeding it to a mouse that lack it and see if we can rescue that. But in the process, when we added the human gene, and now we created a mouse that has twice the normal of the protein, one from its own gene and one from the one we added, we got a mouse also with a progressive neurological disorder. This mouse had seizures, it had learning and memory issues, and it also had very unusual stereotype behavior. And this was really a surprise because this mice just had twice the normal level of the protein and had all these devastating features. So this told us gene therapy is really not an easy thing to do in this syndrome, because if you're delivering the gene through viruses, it's very hard to get the level just right and not to increase it. And if we were to look now at the mice with the doubling, they also had a progressive neurological disorder and premature death. But having observed that doubling the gene is also toxic neurologically, made us go back to the clinic and think, are there patients who actually have duplications of this gene that will lead to having twice the normal level? And I share this story with you because, as I mentioned at the beginning, starting with a human will eventually teach us some biology, but also starting in the lab will actually help us maybe diagnose some disorders and understand them better. And this is what happened in this case. When we went back to the clinic and said, if there are patients who are presenting with autism or Rett-like syndrome, and we look at their DNA and we sequence the DNA, the gene is normal, but how about if there's an extra copy? And indeed, we found that. We found that there's duplications in males, and these can cause autism, Rett-like syndrome, and cognitive deficits. And also this disorder is lethal in the male, but not as early as losing the protein. These males will die by 20 years of age. And on this video, you will see one of those males. He presented with autism. He had recurrent stereotyped activity. And after presenting with autism, he continued to progress to have severe neurological problem. And unfortunately, he is now bedridden, require a lot of respiratory support. And when we looked at his family, you'll see here, this is the young boy I showed you. His maternal uncles all had this duplication, so did his cousin. And all of them, unfortunately, died by the age of 20 and told us that this disease is truly progressive and lethal. But you also notice that his mother is a carrier of this duplication, so is his grandmother. And while they appeared healthy, where they could have a family and carry on with life, they actually had their own neurological problems. Most of them suffered of anxiety and depression and OCD. And these features happened before the birth of an disabled child. You can argue that perhaps having a child with developmental disability can cause depression and anxiety, but these women had all these symptoms before the birth of their child, which told us that even perhaps very few cells, because these women are mosaic, having the duplication is bad. We then identified some patients who had random X inactivation, and they had mild Rett-like phenotype from the duplication. So if I was to summarize then data from this study and data from other studies, what we learned is the brain is very sensitive to the levels of this protein. You have to have it just right. If you have it at normal 100% level, the brain function is normal. But if you have twice the normal level, as in the duplication, then it's severely compromising. And we then found humans that have triplication of the gene, so 3 times the normal. These are even more severe. These die as infant. And when we made mice with the triplication, they're also a lot more severe. And of course, I mentioned to you, if you lose the protein, it causes neonatal death. And in females that are mosaic, one gets Rett syndrome. So now we have models that have twice the normal level of the protein. We have models that lack the protein. We can begin to see what happened in brain cells, what's happening to them at the level of physiology, how their activity is, what happens to them at the level of gene expression, and eventually we're going to look at behavior. So when we look at neurons, these are hippocampal neurons from mice that totally lack the protein, or mice that have twice the normal level of the protein, you can immediately see that they have different behavior. They are inverse of each other. If you lack the protein, you have decreased excitatory current, decrease in their excitation properties, whereas if you have twice as much, you have increase. And when we asked why is that, when we looked at the synapses in these in neurons from these mice, you can mark these synapses with special marker, which you see on this hippocampal neuron. And this is a healthy mouse. You'll see a very nice number of synapses. We found that mice that lack this protein have decreased number of these excitatory synapses, whereas mice that have too much of it have too many synapses. So this taught us one thing very important. It told us that this protein is not important for making synapses. You can make it in the absence of this protein, as you see here. But what this protein is important to is to really dial the right number of synapses for the brain cells to function well. And if you have too little of it, somehow you don't make enough of them, and if you have too much of it, you make too many of them. And the important thing to know also, having too many synapses is not good. Even though the animals seem to learn very well at the beginning, they eventually deteriorate and have learning and memory problems. So the next thing to ask then, what does this mean? What happens inside the cells? How the gene expression changes? I mentioned to you that this protein was predicted to regulate gene expression. So we then took neurons from the mice that either lack the protein or the mice that have twice as much of the protein and asked, what's the profile of the gene expression in these mice? And what we found, they're exactly the opposite of each other. And this is shown here. So if you have too much of the protein, many of the genes go up, whereas if you have too little or none, you see the same genes go down. There were some that were not shared by the two, but quite a few, about 2,500 genes, were shared and they were inversely regulated. And this was true for the hypothalamus shown here, but it was also true for the cerebellum, and it was true for the amygdala. So this is a phenomenon that happened in all brain regions. And what was important to note, although this protein was believed to repress gene expression, what we found is really when you have too much of it, genes go up, and when you take it away, genes go down. So it didn't quite jive with the idea this is a repressor, but we had no idea why these things are happening. We began to look at these genes. Most of them are changed by maybe 20%, small fold change. But we found about 50 or so that are changed by 30 to 50%. Particularly, they were about 25 changed by 50%. And when we looked at those more carefully, we found many of them cause autism and intellectual disability in isolation. So here are some genes that are altered in the Rett model by 50%, either decreased by 50% or increased by 50%. Each one of these genes on its own, if deleted in the genome by one copy, so it's decreased by 50%, or if it's doubled, you're going to have autism or intellectual disorder and sometimes other features. And you can see many of those have been shown causative of such disorders. And imagine that one of them can give you autism and intellectual disability. Now all of these together are altered in the Rett brain. And that will give you an idea how devastating the loss of this protein is. So finding these genes and finding that it's really a daunting problem. And when we knew about 2,500 genes are affected, we knew if we continue to pursue the approach of all these genes being altered in the absence of this protein, it's not going to be a very easy way to solve this syndrome. So where do we go from here? We figured maybe a different way is let's go back to the patients, think about their symptoms, and maybe think about localizing where each of these symptoms come from, and see if we can find changes, identify the cells responsible for the symptoms, and if we can identify them, learn about the properties and the key chemicals of these cells, and perhaps come up with a therapy that modulate that. And this is something you can do in mice. We can't do this study in humans, but what we can do in mice is engineer mice so that we can specifically delete this gene responsible for Rett syndrome from specific type of neurons and ask if we take it out from, say, the neurons that make acetylcholine, what symptoms will emerge, and so on and so forth. And we've done that for all the cells I'm showing on this slide, but today I'm going to just tell you two examples to show you one, sometimes you learn something you had no idea is going on, and the other that proved to be of clinical relevance. So the first of these knockouts we made was deleting the gene in very few neurons in the hypothalamus. These are called the PVN neurons and SON neurons. And when we deleted the gene just from these neurons and we looked for all the features we see in the Rett mouse model, we only found two features. We found anxiety. These animals had a very abnormal stress response, a feature we also see in Rett syndrome. But they also had aggression. This you don't always see in Rett syndrome, and we'll come back to this point. And they also were overweight. The animal were overweight. When we then did detailed studies to find out why are the animals overweight, we learned that when you don't have this protein, you have no control over eating. We found that the animals ate uncontrollably. They didn't know when to stop. And then we did many studies, and we were able to show that this protein is important for regulating the hormones that give us the signals that we have enough food and there's satiety. So this is something we really had no idea was happening, because in Rett syndrome, the protein is lacking in all throughout the brain. And so these girls have so many additional problems. They can't even get to the table to eat. They're in a wheelchair typically. They grind their teeth. You actually take 2 hours to feed them a very small meal. They're very, very affected. So you're not going to see features of somebody overeating because there are many other brain regions affected that are masking these features. It's only when we took this gene from isolated cells, we uncovered a role of this protein in feeding behavior and in social behavior and how we deal with novel environment and aggression. And that was quite insightful. So we published the paper in one of the Cell Press journals in Neuron, and quickly we started getting emails from parents who've made the observation that their kid had this problem. So I'll show you this example. This is a girl with Rett syndrome. She has a mutation in this gene. But unlike a typical girl with Rett syndrome, she was slightly better off in that she could get up, go to the refrigerator, and choose to eat and feed herself. And her problem was eating uncontrollably. And that her mother then finally had an explanation for that. So that was consistent with what we've seen in the mouse. We then got an email from another family where there were two brothers who also presented the hyperactivity, autism, and mild delay, but at 4 years of age, they began to eat uncontrollably and became massively obese and due to hyperphagia, and also had aggressive behavior. The mother said they don't like change, and they actually had attacked so many teachers and students that one of them had to be homeschooled. And what we found is these two boys had a very, very mild mutation in this protein. This is the kind of mutation that sometimes you say it shouldn't really have much of a consequence, but this very, very mild mutation clearly affected some of their hypothalamic function, and it told us that these hypothalamic neurons responsible for aggression and feeding are very sensitive. And these are the 2 young boys that were affected. So again here, some studies that we really totally unpredicted, studying a gene in specific neurons, taught us about its role in feeding behavior and aggression, and brought up some variants on this syndrome, people with milder mutations who are now manifesting these partial features. The next story I'm going to tell you is when we took this protein from a population of neurons that make GABA, the neurotransmitter GABA, and these type of neurons are typically inhibitory neurons. So in the brain, they're excitatory neurons, and these make about 80% of the neurons in the brain, and they typically project within their area, but they also project long distance within the brain. And they carry this information flow along these long projections. The inhibitory neurons, on the other hand, they're fewer. They make 20% of all the neurons in the cortex, for example, and they primarily project locally, and they are modulating the activity of the excitatory neuron. So in this little cartoon, you'll see here 2 excitatory neurons sending some projections long range, and there is 1 inhibitory neuron here synapsing on them, and when there is increased inhibitory signal, you can see it puts the brake on the excitatory neurons, dampening their activity. So although the inhibitory neurons are only 20% of the brain cells, they really are powerful because they're the one also modulating the flow of information from the massive number of excitatory neurons. So what happens if we now took the Rett syndrome gene just from those inhibitory neurons? When we did that, we found that we reproduce practically almost all the features of Rett syndrome. This was quite surprising. We did not expect to see as many features reproduced because from all of our additional cell-specific studies, we usually get 1 or 2 symptoms. But here we got almost all the symptoms, including this 4-paw activity, the ringing of the paws, including a lot of repetitive stereotyped activity to the point where the animals scratch the skin of their neck because they're constantly grooming and itching. And when we looked to what happens in the absence of the protein in the inhibitory neurons, we found that there is decrease in the activity of these neurons as far as the inhibitory current that comes out of them. And we found that that's really due to decreased GABA levels. I told you GABA is the neurotransmitter made in these neurons. So this was quite important because it taught us many things. It told us that loss of this protein, either directly or indirectly, leading to reduction in GABA levels. It also told us that if you reduce GABA by about 30 to 40% in all neurons, you're going to have a variety of symptoms, all those of Rett, and that's pretty devastating. It highlighted the importance of these neurons. So then we asked, maybe if we took it out from the 20% of neurons that control the other 80%, it's no surprise it's going to be so terrible. And of this really to be clinically of value, we need to see what will happen if we put the protein back only in the inhibitory neurons. So now it's really lacking in all other brain cells. And this is work that's just now being completed and done by Kirsten Ury, whereby she only added the gene in those inhibitory neurons and evaluated the animals. And I'm going to just show you a couple of examples of behaviors we've done. This is what we call a nesting behavior, where you give the mouse a little piece of cotton, and the mouse will immediately shred it to make a nice nest. Of course, for the mouse to do this, it has to have 2 features, 2 abilities. It has to be able to use its front paws, and it has to also have the social interest in making a nest. And we can score, this is a very poor nesting behavior. The score is 0, the mouse can't do anything. Here is making a small nest, 1, and a perfect nest would be a 3. Another thing we can test is coordination. We put the mouse on a rotating rod, and if the mouse is coordinated, it can stay on the rod, but if it's uncoordinated, it will fall. So we can record the time it can stay on the rod. So what happens if we bring the gene back into just these GABA-making neurons? You will see here, healthy mice have very nice nests, make a perfect score of 3, but if the mouse totally lacks the Rett protein, it it does a poor nest. And this is just a control mouse here. And this is the mouse where we brought the gene only in those GABA neurons. It's lacking on all brain cells, but it's back only in that. You could see it corrects the defect. They look very much like normal. This is 24 hours after we gave them the nest, and 48 hours. Again, the rat mice still do it poorly, but this mouse rescues back to normal level. What happens to balance and coordination? Here is you see the rat mouse doing very poorly. It falls off the rod pretty quickly, whereas the healthy mice and the control mice can stay longer on the rod. And in purple, you see the mice that have the gene only in inhibitory neurons, again, rescued and doing very well. What happens to survival? Rat mice, as I told you, the males die, but if we add the gene in inhibitory neurons, they live much longer, and we have now that are mice that are older than a year. So this was quite exciting because it told us really if you can restore the function of inhibitory neurons, that indeed is rescuing many of the features of Rett syndrome. So in this particular case, what we've learned from this study is that moderate reduction in GABA signaling can cause a lot of neuropsychiatric features, because that's what happened when we took the gene just from these neurons. And this makes us predict that even a more moderate, more subtle reduction might cause other psychiatric phenotypes, and that's remained to be seen. But what's important, bringing back the function of the protein in these cells can rescue many deficits. Now, the main defect when you take the protein out of these cells is reduction in GABA, and there are drugs that can enhance GABA levels at the synapse. So the next question, and this is why I told you I'm going to tell you a story that potentially is clinically useful. The next question is, can we use now drugs that can enhance GABA level at the synapse or potentiate the function of GABA by affecting the receptors that respond to it? And these are studies that are currently ongoing. A drug called vigabatrin, which is an anti-epileptic drug, which curiously has never been used in Rett syndrome, but it is helpful for epilepsy and has been used in humans before, can inhibit the degradation of GABA. So the idea, if we give this drug, we can boost GABA levels at the synapse, and we might be able to help these patients. And this is a study that's ongoing currently. So the point of these studies is sometimes by breaking down the studies to single cell type, you might learn which neurotransmitters may be driving many of the symptoms of the disease, and you can at least begin to test pharmacologically if a treatment could potentially reverse some of the symptoms. Now, in the next section, I'd like to tell you that we still haven't been able to really understand exactly how the loss of this protein is causing the progressive nature of the symptom and what is really the function of the protein. And to understand a little bit more about the protein, we went back to the patients and we asked, can we learn something about the protein from patients? And the point is, if you study males, you don't have the confounding effect of mosaicism that we see in the females. And if you study mutations that truncate the protein at different pieces, you can find a mutation that keeps most of the protein intact, then you keep adding more and more deletions based on different patients to really see if there's any particular portion of the protein that's really important. And when we reviewed this, we found that some of the mutations that truncate almost half of the protein away, you lose half of the protein, are very severe, they cause death. But some that make them a little bit later in the protein, so you preserve a little bit more of the protein, those may live into years. And what was most striking is right here. You see a mutation that truncates the protein at amino acid 270, and they die by almost 1 year of life. And here's another mutation that truncates the protein 3 amino acids later. So just 3 amino acids. If you're reading a sort of a book chapter, think about this as having 3 words less lost from the chapter, and that makes a huge difference. So this was really curious to us, and we thought, could there be something important here? Now, there are multiple patients with this deletion, but there was only 1 patient with this deletion. So we couldn't conclude that from human studies. We had to really go back to mice and put the same mutation in mice and ask, do we see a difference between the two? Because then that will nail it, that really those 3 amino acids are important. And when we did that, we found indeed this is the case. This is what a healthy mouse looks like. This is a mouse that totally lacked the protein. This is what it looks like. You see it has disheveled fur, it's overweight, it doesn't look healthy. This is the mouse that has the truncation. The protein is truncated at amino acid 270. The protein is normally 480 plus amino acids. So you'll see here this mouse doesn't look healthy. And this is the mouse that just have 3 more amino acids than this one, and it's looking much, much better. And when we look at behavior, we found it the same differences. And here looking at survival, you see the 270 and total loss of the protein look the same, but the 273 looks much, much better and lives longer. So what's different between those two? That really, this particular observation that started with patients led us to go back and look at the protein more carefully and look where are these 2 mutations happening. And what we found is that the 273 happens right here. This is where you truncate the protein, whereas the 270 truncates it right here. And when we asked what's in this region, and we did analysis by comparing this amino acid sequence to all proteins in the genome, we found that this is a region of the protein that has similarity with a family of proteins called high mobility group protein A. And that domain is called an AT hook because it's a part of this protein that make it attached to the DNA when there is the base pairs A and T, AT-rich DNA. So it allows it to bind to the DNA. And these proteins, typically proteins with AT hook domains, such as the HMGA proteins, they require 2 AT hooks to bind. So we knew our protein had first 1 AT hook, and now we discover a second one. So it's behaving very much like this family of proteins. And usually when it binds the DNA, it bends it and allows gene expression to be modulated. So we went on to ask what happens to chromatin. Chromatin is the combination of DNA and proteins that wrap together, and that's usually what changes when genes are being modulated to be either silenced or expressed. So we began to look at markers to evaluate chromatin. And one of these markers is a protein called ATRX that happened to be mutated in another intellectual disorder that normally interact with MECP2. And you'll see that they interact nicely together, even in the presence of the mutations. And this protein was known to be lost from the DNA when we lack the protein, when we lack the ret protein. So now we asked, what happens in brain cells with these 2 mutations? This is a brain cell, and this is a marker for components of the DNA of the neurons where there is DNA that's compact, and that's where the ret protein binds. And you see the protein, our 2 mutant proteins can bind the DNA. And when we look at this other marker, ATRX, we found that early on, when the animals don't have any symptoms, we see the neurons healthy with this area of condensed DNA still present, and this protein is binding very nicely, and you see it in all the mutants. But what we noticed that when the animals are 7 weeks old, and that now the mice that lack the protein or have the earlier truncation, they're now symptomatic, we lost the binding of this protein to the DNA, but still in the less severe animal model. And only when these animals become symptomatic, we begin to lose the binding of ATRX to the DNA. Now, why I'm telling you all this? Just to tell you that this kind of work through a more, detailed experimentation with different mutations led us to a first marker that correlates, that explains to us that something is happening to the chromatin that explains why the onset of this disease is delayed. This was really something perplexing us. Rat girls look normal for a few months, sometimes up to 2 years, and then develop symptoms. The mice look normal for 4 weeks, then develop symptoms. And we had no handle on this. But these kind of studies gave us a handle. This doesn't tell us this is what's causing the disease, but it gives us a handle to begin to look to what's happening to the chromatin and how it is changing. And you'll see here, this cartoon shows you what the normal protein looks like. It can bind the DNA, and these are the 80 hook domain that sort of help bend it. If you have the truncation that's milder, you can still bind. It's not perfect, but you can still bend the DNA. But the truncation that's at 270, you totally lose that. And of course, if you don't have the protein, you lose all of that. So this is now beginning to allow us to design experiment to really understand, when does the bending happen? Does it happen when the neuron is stimulated, or does it happen when the neuron is silent, or does it happen all the time? These are the kind of questions we're now pursuing to understand why this protein is so essential for every neuron. So what I told you so far is that we created mouse models that are helping us understand behavior and eventually do preclinical studies. We've also looked at chromatin. We were looking at that. I told you what's happening to the synapses. The one thing we have not talked about, and that's still work in progress, is neurons are not functioning in isolation in the brain. They actually function in a circuit. They connected to each other. The circuit functions together and can affect other components of the brain. So we've decided it's really important to understand this disease, to look at the circuit. And in particular, we wanted to do this for this important clinical reason. You'll see the same theme that always, it's observations in the patients that's making us rethink what's really going on. I told you about Rett syndrome, and I told you about the duplication syndrome. What you'll notice is that most of the features are the same in both syndromes. Most of them. There are few that are different. You get small heads in Rett and some autonomic problems. You don't see that in the duplication. And you get a lot of recurrent infections and hypotonia in the duplication. But all the major features are very similar. And in fact, the patient can be mistakenly diagnosed. But if you have been following, I also told you, when we look in the cell property, they are the opposite of each other. One has decreased excitatory synapses, one has increased. If I told you the gene expression, they're the inverse of each other. So how is it that you have a behavior that's finally in the human the same, but you're having molecular and physiological properties that are totally the opposite of each other? It was actually this observation that made us begin to think we have to look at the circuit. And Lou in the lab has started these experiments. And how do we look at the circuit? In this particular example, I'm showing you the behavior of a type of neuron in the hippocampus called the CA1 neuron. And we can sort of sneak on these neurons and watch them within their circuit, how they're behaving. We can watch their activity and record it. We can do that by physiology, which we've done, but you can also do it by imaging. You can use a calcium indicator, which is very sensitive. This calcium indicator the cell will fluoresce anytime there is increased calcium in the cell, because the cell is now active. And I want to show you just what this looks like if you look at a healthy CA1 neurons in their intact circuits. From a healthy mouse, you'll see few cells firing. This is spontaneous. We're not stimulating, but this is how these brain cells are firing. So when we look at that in the mice that either lack or have too much protein and look at the number of times they fire, we find that the ones that lack the protein fire less, tend to have fewer cells that fire, and the ones that have too much fire more. So everything we've seen at the cell level is still the same. Now what happens if we look, okay, they fire less or more, but how do they fire in a circuit? How do they behave together? We begin to ask how many times they fire together. What's their synchrony like? And that's where the surprise came, and where we find the two mutants look very similar. So here's again, look at the top video. You'll see in wild-type mice, few cells are firing, but you'll find one fires at a time. They're not firing together. I want you to look now in this model, in this duplication model, and you quickly see that the cells tend to fire together. They're almost in concert. We call that hypersynchrony, and they have much higher coefficient, correlation coefficient. And we find that the same thing is happening in the knockout. So finally, we have a circuit behavior, which is that of increased synchrony, where the cells fire together more, rather firing more spontaneously at different times within the circuit. And we see that both in the loss and in the gain model. And we're now exploring what's driving that hypersynchrony in both models. So, in summary then, to review all that I've shared with you today, we've learned that mutations in this gene can cause Rett syndrome and a variety of neuropsychiatric disorder. I did not tell you the story about the importance of this protein throughout life, but we know that you need this protein throughout life, which means if you let the animals grow with the gene and you take it from the brain in adulthood, the animals will develop Rett syndrome. So that's important, because that means any treatment we discover has to be safe so that it can be used chronically. I mentioned to you that deletion in select neurons is affecting their function, and now we know in part by decreasing the respective neurotransmitters. And in the case of GABA, we're hoping if we restore the level of that neurotransmitter, we might improve some of the symptoms. But also, what's really important is from a neurobiological point of view and clinical point of view, we can now exploit this strategy by taking the protein from different cells and begin to assign behavior to specific neuronal subtypes and specific neurotransmitters. Lastly, study the circuit dynamics in these disorders we're hoping will explain to us the final outcome, which is the behavior, and that's what's still ongoing. So in some ways, our journey with Rett syndrome has really paid off in the sense that we are at a time we're contemplating therapies that we hope will be, there's no guarantee this will work. We did the genetics to really be sure at least it's worth trying, and we hope that some of this will translate to effective therapies. Of course, I'm grateful to all the people in the lab who've contributed to this work, particularly Ruthie, who cloned the gene, and the team of investigators who've made individual knockout, and the recent work that's ongoing from we on the circuit and Kirsten on the rescue experiment, and many collaborators, and of course the families who've trusted us to really share DNA samples and continue to come back for a visit and help us study them. And of course, it's very hard not to say it's been a most rewarding experience, as devastating as it is to see a child with Rett syndrome. At least it is rewarding that we can diagnose these girls earlier now, whereby physical therapy and some behavioral therapy and interventions are giving, and nutritional therapies are giving them much better outcomes. We don't see them as much in wheelchairs anymore, so that's really helpful. And of course, I'm very grateful to all the people in the lab who've done this work, and to all of you for inviting me to share this story with you. It's an honor to really be invited and rewarded for doing what I love to do, which is help these patients and help understanding their disease better. Thank you.